and other physiologic processes. During the processing of painful inputs, noxious stimuli detected in the periphery are transduced into electrical activity in primary sensory neurons called nociceptors. Activated nociceptors transmit neural signals from the periphery to the spinal cord, where they release excitatory neurotransmitters such as glutamate and substance P. These neurotransmitters activate second-order neurons in the spinal cord or brainstem, which carry the pain signal up to the brain. Endogenous opioid peptides modulate nociceptive signaling all along this neuroaxis, from the periphery sensory neurons to higher brain areas. Various peptide ligands preferentially bind to three types of classical G-protein coupled opioid receptors, namely enkephalins, which interact with delta receptors, dynorphins with kappa receptors, and endorphins with mu and delta receptors. Opioid receptors are expressed throughout the central nervous system. In particular, high concentrations are present in the limbic system, hypothalamus, medial thalamus, brainstem, and spinal cord, all of which function to integrate nociceptive signaling. Opioid receptors are also concentrated peripherally in the gastrointestinal tract, where their activation reduces motility and increases fluid absorption. Looking more specifically at the analgesic effect of opioids, mu-opioid receptors in the substantial gelatinose of the spinal cord are thought to play a particularly important role. These receptors are located both presynaptically and postsynaptically at the first central connection between the first-order nociceptors and the second-order neurons that carry pain signals up to the contralateral spinal cord to the brain. Presynaptic receptor activation inhibits the release of excitatory neurotransmitters, whereas postsynaptic ligand binding reduces the responsiveness of the second-order neurons to excitatory inputs. The result is a dampening of incoming signals generated by painful stimuli.